All right. Hello, everyone. My name is Hasnain. I'm a biology major and I'm an undergraduate as part of Dr. Sladek's lab. And today I'll be presenting my completed research on the comparison of soybean oil and olive oil-based diets on expression of colonic HNF4 alpha. Okay. So let's first talk about soybean oil. So soybean oil is the most consumed oil in the US diet, and it's found in virtually everything. So this is from bread to meat, cooking oil, fast food, processed food, literally everything has soybean oil in it. So the thing about soybean oil is that one of the most prominent components of that is an acid called linoleic acid. Now, linoleic acid is an omega-6 polyunsaturated fatty acid, and it makes up around 55% of soybean oil. So as you can see in this graph here, for soybean oil, linoleic acid takes up a huge quantity of that. Now, we'll be talking a little bit about olive oil too. That's another oil that I looked into. Now, olive oil is largely considered to be a healthy oil. It consists mainly of oleic acid, which is a monounsaturated fatty acid. And oleic acid takes up almost 3/4 of what olive oil is supposed to be, and has a smaller fraction of linoleic acid. So over here, we can see the buildup of each of the oils, and then over here, we can see the structural compounds. The main difference is the presence of one double bond. So that's all you have to know about soybean oil and olive oil. I'll now be discussing inflammatory bowel disease. So incidence of inflammatory bowel disease is on the rise. So basically, what is inflammatory bowel disease? It is basically known as inflammation of the gastrointestinal tract. So what that means is that basically inflammation of anything from the esophagus to the end, like the colon, the rectum. So there's two types of inflammatory bowel disease. So one of these is Crohn's disease. Now, Crohn's disease can involve any part of the gastrointestinal tract. As you can see here in this visual on the right, we have some redness, and that shows where Crohn's disease is likely to show, some in the small intestine, some in the large intestine. And then the other kind is colitis, ulcerative colitis, which is the inflammation is mainly going to be seen in the colon. So as for inflammatory bowel disease, it has been steadily on the rise, and along with that, consumption of soybean oil has also been on the rise. So as you can see in this graphic over here, soybean oil ever since the 70s really has been spiking. And this graph over here shows the million pounds consumed. So just in 2015, we hit 20,000 million pounds. So soybean oil and inflammatory bowel disease are showing to have potentially a correlation. So there's many genes that are linked to inflammatory bowel disease, but one of these is called HNF4 alpha, which will be one of the main parts of what I'm presenting about today. So HNF4 alpha, what is HNF4 alpha? So it is basically, it's a highly conserved transcription factor. It stands for hepatocyte nuclear factor 4 alpha. And basically what it is, it's an IBD susceptibility gene, IBD meaning inflammatory bowel disease. And what it means is that interestingly, it binds to linoleic acid. And in this process, it is also seen to be linked to colitis and to diabetes as well. And within HNF4 alpha, there are two promoters, which in turn power two different isoforms, so P1 and P2. Now, on this graphic on the right here, you can see that there is P1 and P2 here. The difference is that there's around 16 to 30 amino acids, so P1 is a little bit longer. So these promoters are slightly different from each other, and they can be seen in many different ways. And as for where it's expressed, so P1 is typically expressed in the liver, whereas P1 and P2 can be expressed in the intestine. But the important thing to know about HNF4 alpha is that it's found in almost every animal organism that there is. So for P1 and P2, P2 is seen more at the base of the crypts of the colon. So here in this graph, we can see this is a cross section of the colon. So P2 would be found towards here, whereas P1 would be found towards the tip. And in this graphic in the middle, P1 would be found up here towards the tip, and then P2 where this red coloring is. So we have seen before that a diet with soybean oil can show a decrease in the expression of P1 and an increase in the expression of P2. And we've also seen that mice that only express P2 are seen as more susceptible to colitis. So now I'll be discussing the actual makeup of my research, which is the research question. So what is my question was, what is the impact of these diets on the expression of P1 and P2 HNF4 alpha in the colons of these mice? So I hypothesized that mice on soybean oil diet will show the highest signs of colitis. And I wanted to see less change in HNF4 alpha with olive oil as compared to the soybean oil diets. So now I'll be talking a little bit about my methods. So for the methods, what I did for this, I've been working on this for months. So I took 10 mice and I grouped them into 3 different diets: olive oil, soybean oil, and vivarium diets. And you can see on this table on the left, I included some information about the percent of calories that come from linoleic acid and the percent of calories that come from fat. So as we can see, vivarium is the control diet. It doesn't have much fat in it, only 13%, whereas olive oil and soybean oil have comparable amounts of fat, but they do vary with their percent of calories from linoleic acid. So this is an important table to just note the different buildup of each. And over here, we can see the average US diet shows about 8 to 10% of kilocalories from linoleic acid and 33 to 37% of kilocalories from fat. So the diets that we were able to obtain in this are very similar to what the average American eats. So over here, once I split them into groups and I put them on diets for 10 weeks, I would feed them and I would weigh them. And then afterwards, I did this treatment called DSS, which stands for dextrin sulfate sodium. Now, dextrin sulfate sodium is known to induce colitis-like symptoms. And basically, it's a chemical compound that when mixed in the water and when the mice drink it, then it essentially scrapes their intestinal lining, which is known as the epithelium. So once it does that, then it is able to show a lot of colitis-like symptoms, which I'll be talking about later. That is what I'll be discussing with the disease activity. So after that, I did that for 6 days. On the 7th day, I euthanized them and I collected their tissue samples. And then I analyzed their disease activity. And I also ran a Western blot, which I'll also be explaining a little bit later. All right, so these are the first phase of the results. So as you can see on this left, this is the daily food per mouse. So we basically measured the amount of food that the mice would eat. And you can see in this graph that vivarium mice ate significantly more, whereas olive oil and soybean oil ate less. This is because the vivarium diet, it's the control diet. There's not much energy in it. There's not much fat in it. So mice will need to eat more to compensate for that lack of energy. And these asterisks and these notations here are showing that the vivarium diet, the results that I got from this are significantly different than the numbers I got from here. So it shows that there is definitely a difference in these high fat diets versus the vivarium diet for the amount of food that they ate. Now on this right over here, we can see that this is the percent change over initially when they were put on the diet. So this is how much the mice gained their weight in terms of like percent increase compared to when they started. So as you can see here, initially they all started the same and olive oil and soybean oil were not easily distinguishable. But then towards the end, you can see that soybean oil, olive oil, and vivarium diets all had very distinct numbers, and soybean oil showed definitely the highest percent change since the beginning. So next, I'll be discussing the disease activity index. So what is the disease activity index? These are basically parameters that we use to judge the disease factors. So for colitis, this would mean fur appearance. If a mouse had more ruffled fur, that is a sign of colitis. Activity, if a mouse was more sluggish, more lethargic. Prolapse rectum is when a part of the rectum is poking out of the mouse, so that would also be seen as a sign of colitis. Fecal consistency, so any diarrhea or looser stools would be seen as a sign of colitis. Hemoccult tests would be testing any blood in the feces. So that is also an important thing that we looked for. And then blood in the cage if there was any additional bleeding outside of that. So on this graph on the right here, this is the disease activity index that we graphed it on the right. So you can see that the soybean oil had significantly higher amounts of the disease activity index. And while olive oil also did have high amounts, both of these were definitely far from the vivarium control diet. So that shows that definitely these high fat diets are causing colitis when they are exposed to dextrin sulfate sodium. All right, so the next part of my project was to be using a Western blot. So a Western blot is mainly used to quantify and to see what proteins are present in a sample. So the way that I did this was initially you have to extract the protein. We did a whole cell extract and we homogenized the samples. We mixed up the colon that we harvested really well and we extracted the protein. We ran something called a Bradford assay, which is supposed to basically show the protein concentration. In this case, we loaded 50 micrograms per 50 microliters. We ran a gel, we separated proteins, we transferred, and then over here, when we detected the protein on the blot, we probed it for different antibodies. So here we were probing for P1 and P2, which I'll discuss. And then afterwards, we developed it and visualized it. So next, what we can see here is the Kumasi stain. So really quickly, before I get into the actual blot, the Kumasi stain's purpose is to make sure there's been no loading errors, make sure that there is protein on the blot. So as you can see here, this band right here, which shows like a decent amount for all of them, this shows that there was no issue with the protein loading. There is protein on the blot. But the most important thing to note is this right here, which is basically the distal colon whole cell extract probe with P1, P2 antibody. So we took this blot and we put it in a solution that contains both P1 and P2 mixed together, and we're going to try and look for them at the same time. So in doing that, we had two positive controls. So these are two different samples that we know will have either P1 or P2. And we do that to use that as a reference so that when we compare our actual samples and we can have something to look at. So over here, once we did that, we probed it with P1, P2, then we saw the results. And HNF4 alpha is seen at around 40 to 50 kilodaltons. So that is the ladder over here. So it's around this band. So what you can see here that's really significant is that vivarium expression of P1 and P2, you can see it. But once you get to olive oil, it decreases a bit. And then once it gets to soybean oil, it almost is completely unnoticeable. And what that means is that there's definitely something that's present in the soybean oil that is causing the expression of P1 and P2 to decrease. There's something in there that is in the soybean oil mice colon that are manipulating the expression of P1 and P2. So that itself is enough to prove that soybean oil does have a correlation with what I'm studying. So the conclusion of this was that mice that were on the soybean oil diet, they showed the greatest susceptibility of colitis. And this was because of the decreased expression of HNF4 alpha, as you saw on the previous slide, where soybean oil saw almost nothing come up on the bands. And as far as where I want to take this in the future, so I also wanted to look into this other marker called LCN2, that stands for lipocalin-2, and that is known as a fecal inflammation marker. So we will use samples from the feces and we'll look into if there was any bleeding present. And then another thing I wanted to look into was HNF4 alpha balance and the soybean oil presence of that. Since we only saw that soybean oil kind of decreased both, I wanted to look more into that and see if soybean oil would show more of a decrease or more of an increase in P1 or P2. And I wanted to compare that further with olive oil. Instead of seeing a decrease of it, P1 and P2 as a whole, I wanted to compare P1 and P2 with each other. So that's some future places that I would want to take this research with. So as for acknowledgements, this project would not be possible without Dr. Francis Sladek and Dr. Poonam Dial. Both have helped me greatly. They have mentored me, they have guided me, they've spent countless hours with me troubleshooting and basically helping me with anything. So I really appreciate their help. I also wanted to give a huge thank you to the members of Sladek Lab, researchers and undergraduates, Gary, Shraddha, Varad, Ritika, John, Gayatri. Everyone has helped me so much indirectly and directly with this project. And lastly, this project was funded by the UCR Undergraduate Mini Grant Program, and it would not be possible without them. And here are my references, and that'll be all. Thank you all for listening.